Okay, here I will quickly show you a few examples. People use ChIP-seq or ClIP-seq or whatever we call. The first one is about the transcription factor study. And the first ChIP-seq paper was published in Science in 2007. And they identify a transcription factor, neuron restrictive silence factor. They want to, this factor, this is a zinc finger repressor, the negative regulate many neural genes in stem cell or progenitor cell or non-neural cell. So they want to identify this binding site, this factor's binding site in the human T cell lines. They use the technology Illumina, but because they say it's only in 2007, they used the 25 base pair reads. They have two conditions with a biological replicase, and the reason two can I don't remember is two conditions, or could be one is the chip, one is the control, and from this one. And I like your question, like why this pig means, that means she was thinking about everything. So I hope everybody can think about when I talked about this technology or this pattern or something. You can say these are binding site compared to control, and no binding site, no random sequence showed here. So we have a high confidence, or which means p-value is significant for this binding site. So in genome-wide, and the author, they identify about the 2,000 noses, which is associated with 1,000 genes. And compared to old technology or others, is 5 times more signals. And also, they found that the accuracy by bioinformatics analysis, they find the accuracy is pretty high. They use the one curve, we call the ROC curve. You may use that in future more and more, and we call the receiver operating characteristic, which shows how much false positive and how the fraction of true positive. And you can see the curve, the area under the curve is pretty high, which means the accuracy is very good, or the model is very good. And people already know this transcription factor has a motif like this way. It's a connected, looks like they have two parts, the left side and the right side, but the left side and two sides, so we can see if it's about the 11 base pair from the left half and to the right half side, which we call the canonical motif. People already know. But in their sequence data, when they identify a number of binding sites, and when they do sequence analysis, they found many peaks. Many peaks, they are highly, they try to calculate the distance, half side distance from half side, this right, left half side to right side. They find that most of our 11, it matches the canonical motif. But however, we see some peak here. It's about 16. So what's that mean? It's a little bit means, it means the left side and the right side, they separated a little bit far. And they found that there is no additional consensus sequence in the middle. So they said there is a space here. So it's a new novel motif. They found that this one is very interesting. There has a significant biological function, but I won't talk there. I just want to show you, ChIP-seq is powerful in that it can help you to identify canonical motif and also try to help you to identify new motif or novel motif. Another one is people use ChIP-seq to study the histone. So to study histone modification. This is published in 2007 in the top journal Cell from Dr. Kyrgyzov's lab. And from that one, they use the T cells and they try to identify 20 histone lysine or arginine methylation, histone variation, H2A Z, which is highly conserved through evaluation. And also they want to see the pattern of poly2, because poly2 is strongly associated with the transcription. And they also want to see the pattern of CTCF. CTCF, we already see previous example, they shows of CTCF use ATAC-seq to identify CTCF and motif. Why CTCF is important, and when you do research in future, you will know CTCF is kind of insulator binding proteins. They may separate those regions into insulator or enhancer. It's very interesting. And they use the platform they used is Illumino in 2007. They used the 36 base pair arrays. This is a pattern they already got for each one. I want to talk one by one, just to show a few examples. Here, they have matched the RNA-seq data. They separate the genes into four groups. One is the red one is highly expressed, or medium expressed, or low expressed, or silenced, not expressed at all. What means that it cannot be detected by RNA-seq. So for this one, you can see poly2, which has a high density signal. Here we can say, okay, signal is a kind of, for high expressed gene, we can see high poly2 signal. And for medium one, we can see medium signal. And for interesting is even for silent one, we still can see a little bit of signal here. But in general, poly2 signal is quite correlated with the gene expression. And also, they try to see some active signals, active modification, and we can see very similar to gene expression, but I won't spend much time here. Otherwise, you may kill me. Next study is people use CLIP-seq. Actually, it's from Yunlong's lab. They published this paper in, I forgot which year, maybe 2009, yeah. 2009 in Genome Researcher, which is a promising journal in bioinformatics area. They use this one to identify the binding site of one splicing factor, SFRS1. I don't want to tell you how important this splicing factor, but just use it as an example. And they use the cross-link first, and then extract RNA, and then IP, then purify RNA, then do sequencing. At that time, they used the 454 life science large genome sequencing, and to identify about over 20,000 binding sites for this one. And first, they examined the CLIP-seq sequencer length. They found most of them, blue one here, most of them, the sequence length is from 40 to 80. It was expected. And as a comparison, the input sequence, they show signal in the 40, but not here. And they identify the motif is a GAA, which is very similar to people already know for this transcription, for this splicing factor. And then, based on the similar to ChIP-seq, they identify number of sequences, and they want to know where they're located. And not surprisingly, because this is a splicing factor, you can see most of sequences, they located on the exon, on exon intron boundary, or exon junction, on intron gene, and but still we found some intergenic region for some reason, for unknown reason. And then they compare this one, but how can you know this sequence they captured is really associated splicing, but we have no idea how to directly validate that, but still, in genome study, they can find the evidence. When they look at, they separate those sequence region into different based on alternative events. They found cassette, or they define the retained intron, and the alternative 5' SS, alternative 3' SS, or upstream adjacent, downstream adjacent to the exon. They found those sequence which has interaction with the splicing factor. They are mostly enriched, overrepresented in the alternative splice side instead of like a cassette exon or retained intron. And you can say this is expected, which means random pick those ones they are assumed to be there. But the observed one is the real one. They identify, they pick up those sequences, they identify their location, they compare with that one. That's what we call observed. You can see this two number is smaller than expected. Then that means it's a kind of depleted. But those numbers are higher than that one. It's overrepresented. So which suggests this sequence bounded by splicing factor, they are associated with the splicing factor. They are splicing events. Another study we can use ClipSeq to identify microRNA messenger RNA interaction. So how many of you have the knowledge about the microRNA? Okay, not too many. Okay, I just go quick through what's microRNA. microRNA, so microRNA, the important, I don't talk about too much about this one, just what's microRNA. You know, after transcription and then we get a messenger RNA, but still we have some small molecules. microRNA, they are usually, mature microRNA is usually 22 nucleotide base, very short. They can map on those messenger RNA. If they match there, by some condition, they can digest those messenger RNA. Then you cannot have a protein product. So even you have tons of messenger RNA, but maybe you get nothing from the protein structure, protein ductor. So this study tried to identify microRNA and the messenger RNA interaction. But you may ask me a question. You know the microRNA the sequence should match messenger RNA. Why can we predict those targets? Yes, we can, but there is a very high false positive rate. The reason is that in human, in animals, in panda is fine. They require microRNA almost must be exact match, so about 22 nucleotide. But in human, in animal cells, the microRNA, the match is not so strictly required. And the microRNA from 22 nucleotide, but most of them, they only require from number 2 to number 8, so only 7 nucleotides. If 7 nucleotides can match to somewhere messenger RNA, especially 3' they may have a digestion. So, but you can imagine, look, only 7 nucleotides. When you match the whole genome, probably you can find the match site probably average 4K genome wide. So you can get tons of region for one microRNA match. So that's a high false positive rate. Now, people, what we can do? So people, they use this technology, clip sequence, try to identify that one because they know the microRNA, if microRNA has to digest this one, they have to work together with the protein they call ARGO. So they use ARGO, and they use this one covalent. They use his clips to covalent cross-link ARGO with both either messenger RNA or microRNA in the same time. Then they sequence them, and if we find some sequence, they have a sequence match from messenger RNA and the microRNA, then we mapping those one to the whole genome. Then we say, okay, those one are target of this microRNA. That's the main idea of this work. Before talking about this work, they did something good for our lecture. We know, and we do sequencing. But usually, and the experimental guys, they always ask the question when they come for us for sequencing, how much sequence depth we need? What we can say? We don't know. We only have a general idea how much we need, but it's case by case. I mean, we must have something we can simulate it, or we have an understanding. Then we can do something we call saturation simulation. What means saturation? For example, here, I don't remember how many sequences they did here. Let's say just 100 million, okay, but it's impossible, probably tens of millions, let's say 100 million. And they have 100 million sequences here. Then they random pick up a partial portion of them. For example, they pick up 16, they randomly pick up 60 million of them. Then from 100 million, they can identify how many microRNAs. This is only based on microRNAs. And then from 16, they try to see how many they can recover. They can get those ones. And this is the rate that they show the curve here. If we say curve goes up, never stop, then we say, okay, it's a far away from situation. Good thing is in this experiment, we can say it goes quickly to be flat here. So which means even if you decrease the sequence depth or enlarge sequence depth beyond the cutoff or threshold here, it doesn't change much. Then we call this is a saturation point. So maybe we say, okay, this sequence depth is enough. I will say that again for ChIP-seq in next few slides. But in this one, they did that sequence. They want to prove their work is good. The sequence depth is enough to do that. And then also they have a 3 triplicates. They also have they try to see how the result correlated with each other, which shows reproducibility. Reproducibility is very good. They use normalized microRNA to see, so each dot is 1 microRNA. So we can say in the one sample what's the frequency, and in the other one in another one what's the frequency. We can see the R square is much higher. It's pretty good. Quality is really good. And again, when they identify those microRNA in the same time, they also identify most of messenger RNAs, and with the matching sequence to that microRNA, you can see where are those messenger RNA come from. We can see they come from mostly 40% of them from 3' UTR, very few on the 5' UTR. In biological, it makes sense because in principle, people think microRNA works on the 3' UTR to digest those messenger RNA to prevent their be produced as protein. And also, when they look at the microRNA, when they look at the microRNA frequency with the matched messenger RNA frequency, we can see the correlation is very good. So that means the bias is not quite obvious when they pick up microRNA, and also same thing with the microRNA target. They have a similar frequency. That's very good, right? And another important thing is that they show here they match together, and each, for example, here, which shows the location, the position of microRNA cis. Again, I show the cis is from number 2 to number 8 nucleotides. Cis, the matching cis determine the target of microRNA. It shows the matching cis located on the peak. On the peak they identified. But for other non-peak area, for very low expressed microRNA, the cis sequence can be anywhere, which could be a negative control. So we have a high confidence about this result. But anyway, in general, from this one, you know the microRNA, the ChIP-seq can use to identify the protein-RNA interaction, and also can identify microRNA target interaction. That's a good thing. That's the advantage of this technology can be used. In 2012, there are a number of papers published in Nature. They use a high throughput technology to do access chromatin landscape, transcription factor binding fire. This comes from a big consortium we call the ENCODE. ENCODE consortium is an international collaboration of a research group funded by National Human Genome Research Institute. The goal of ENCODE is try to build a comprehensive part of all functional elements in the human genome, including elements act at the protein or at the protein level, RNA level, DNA level, how to like all the regulation network. So here is the ENCODE website, and you can see how many species they studied. 4 species: human, mouse, worm, fly. And you can see how many projects they have. They have over 10,000 projects. And also you can see how many serialized samples they come from. And from that website, you can find a detailed one. The good thing in ENCODE is it published open access. So if you have any question, or maybe you can find the answer there about your question about the histone and the splicing issue, you may try to see if you can find any evidence from their published data. Yeah, it's a very useful. Sometimes when we try to understand the mechanism or something, we don't have money to do experiment. Then we go there, try to find the evidence. And also, if you want to know what the work, or if you want to know the method they published, and also you can go to Nature website. They have a specific section for the ENCODE, and you can find a number of papers there too. Okay. I know looks like you are not excited about technology, but still you have to think about what experiment consideration for this experiment. Especially here, we focus on ChIP-seq, because ChIP-seq is broadly used in transcription factor binding, histone modification, or anything. And yeah, what's the most important thing in ChIP-seq? I already showed the slides here. I should ask you before I show here. Yeah, it's antibody, right? If antibody is not specific, whatever you got is messed up. You don't know what's that. You have no idea what's that. So antibody is most important. It's specificity. And another one is amount of initial material. Usually it's not too high, but still. And then here we show and we can use a barcoding, or we call it multiplexing, for small genome studies like yeast. So we can use a different barcode and for different study, and we can put them in the one day, so we can save time, we can save the money, and also we can avoid the bias by PCR because we need a large data for those sequences. And for paired end, which can help us to analyze the area in the repetitive regions. But the question is, do we need to replicate or not? I keep this question until the final slides. Of course, and the most answer is yes, we need to replicate. But I also will tell you why sometimes when we use replicates, we also have to be careful. This is a biological question. And another consideration is I emphasized, for example, and in previous example, we have to be careful to choose a sequence depths. We need something like saturation analysis to find the optimized sequence depth study. So as I mentioned in the ChIP-seq short example, the result of given access do not change when more lists are obtained, which means after that becomes flat. But when we look at the ChIP-seq data, if we said, okay, for ChIP-seq data, we can have a static significance for those ones. We want to know, okay, they are significant different from the control or from background, from noise, from whatever we compared. We have this one. But unfortunately, if we only best this one, it looks like never saturated. It always goes up, up, up. Then one thing is because when we have more depth, the power is more power, so it goes up. But if we add another criteria, like we said, okay, we also want to consider the photo change, enrichment. So how can we do? Then, yeah, that's good. If we have additional criteria, maybe the curve becomes flat somewhere. Then at this point, we can say that could be the saturated point. We can use this one. Okay, but be careful. Whatever you added there, I mean, like a criteria you use for spatial pig calling algorithm, they always have significant impact on the number of pigs you selected. Now you are happy. You can see a formula here. It's not biological. Then we will have a tens of formula in next slides, okay? No response, just kidding. Yeah, and you can see from previous one, I never show the formula. I don't like them too. But the reason I want to show here is quite clear. This formula shows published from Kirchhoff's lab 2 in 2010. They show us how to evaluate the sequence depth based on this one. You don't need to memorize this one, but I want to mention several things here. One is S, of course. What's S mean? S means the sequence, how many total reads we need for that one. It must be at least this number. Forget about the factor here. What's G? G is genome size, non-repetitive genome size. But if we want to say, okay, we study human, it's always fixed there. But what's the W here? W is a window size. Window size is kind of like resolution. See how big area you want to take a look. Then this could affect the final depth you need. And what's E here? E is enrichment. Like in this window size, if you want to compare to background or compare how other things, you may say, okay, I need a 2 for the enrichment cutoff, or I need a 5 for the cutoff, or I need a 10 for the cutoff, whatever you need. And then it will affect the final depth you need. And what's N? N is evaluate the fraction of genome could be enriched, like transcription factor. You say, okay, probably the transcription factor I did probably only 1% the whole genome. That's fine, it's only 1%. But maybe for another transcription factor, you may say, oh, it may be 10%. Yeah, that's 10%. But how they will affect to this one? So let's go from assumption first. This sequence, this formula they derived is they have assumptions, which means non-specifically precipitated text, they are distributed uniformly and can be described by points distribution. Okay, and also all this they have a cutoff. They determine the regions and the enrichment or fractions. They have a cutoff based on cutoff p-value 10 to -4. If we change those ones or change mode, they may change. But still, even the final number is not exactly right, but still it's a very good evaluation. Now let's look at the list. I highlighted this one. And let's assume we only care about the human. We highlighted this one. If window size is bigger, of course, the resolution is lower, then depth is lower. We don't need much higher, much more sequence. But if you want the window size is smaller, that means you need a higher resolution. The depth, you need more depth of sequence. And also, if you evaluate the fraction of genome enriched, like the binding site, you evaluate, okay, maybe only 1% of whole human genome, 1% here, then we may ignore that one. From 1% to 2%, maybe nothing changed. You don't need much care about that. But if from 1% to 10%, I have to say they changed much, probably double or somewhere. And also this E is an enrichment. It's also inversely proportional to the sequence rate. Well, what's that mean? That means if you have a cutoff for the peak you caught, if you said, okay, I need a 2 for the change cutoff, okay, then I say that's not quite much maybe. But if you said I need a 5 for the change and I need or even 10 for the change, then I have to tell you, if you want to identify those two, as most as possible two peaks, you have to much higher sequence depths. So just remember the relation between those ones. That's what I want you to learn. And I learned too. Now, the next question is, if we don't get the saturation steps, is it, how much do we need? Is it good to have as much as possible sequence rate from 1 million to 10 million to 100 million reads? Is it good or not? Who can answer this question? Okay, to save time, I don't ask you again. Yeah, and yeah. Another question is, where increasing sequence steps get more true binding sites? Then how many sequences are detectable around the true binding sites? We have to look at it, because from this one, this is some people, they use the data. Before answer this question, these questions, I show you a simulated data, and they showed what's the data, and they published in about 2010, 2007, 2008. They show something like they identify a number of peaks and the ChIP-seq peaks, and they identify the motif of their carrier body. Then they want to see how many takes around those chips within 100 base pair. And you can see some of them, they may have a much high text, so which means the signal is very strong. But most of them, the signal is not quite strong. So what's that mean? That means the peaks, they have a very wide dynamic ranges and no clear subproportion for binding positions. So then let's go back to the questions here. So if you get more sequence, probably I can expect there are more sequence here, more sequence here. But does it help you to get more sequence here? No, they already identified, so maybe not. And also, what this one means, this is, so they have a very few tags to show the binding site, which means they may be weak binding site, or maybe totally, maybe false positive. So if you have an extremely high risk, you may have a high false positive somehow. So that's the reason why you have to try your best to find the optimized the sequence depths based on your study. Different study may have a different sequence depths, like iSeq, ChIP-seq, ATAC-seq, they are quite different. Also, even the same platform, like ATAC-seq, what do you want observed, what you want studied, they may have a different sequence depths requirement. Okay. Now, let's answer. Increased sequence depth will allow a great number of weak binding positions to be discovered. So be careful. Another thing is if the design is okay, if the platform, I mean, like used is right, so maybe small sequence depth is enough to identify those pronounced binding site. That's what we showed before. And when you ask, do I sequence enough, like a saturation question, we should always ask another question: how weak six binding do I allow? It's like a very weak. So you have to think about it's a balance. You have to think about it. It's like mostly in mode one, we want to know is we need something we call the minimum saturated enrichment ratio relative to the background to understand how much you have. Here is one example to show, and actually it's a little bit complicated. I don't want to make you frustrated, but still, I think maybe show you something in other way. Here they have this is a Yunon size. So here they have a sequence this number. So they claimed, the author claimed, okay, if we reduce the data set to somewhere here, we still can observe the 99% peaks. If we choose the cutoff, if we use the 7.5 for the change in enrichment. Of course, you may say, okay, even I dropped from here to 1/3 of this one, based on this fold change, we still can observe about 90% of peaks. Not bad, right? Let's say that, okay, yeah, it's good. And they also, I think in the same table, they also identified this relation between MSER and the number of tags. It's a similar like a linear log-log relation. So what's that mean? Like the enrichment here, if we want to see twofold minimum twofold change, so we have a 0 here, then we need this number of sequences. If we need a threefold here, like a 2 minus 1 is 2, and probably we need this number of risks. So yeah, it's a beautiful evaluation, but still I have to say a particular value cannot imply all true binding site. Probably we never can get all true binding site with a specific enrichment. It only appears increase the sequence doesn't help too much. It just shows the other way. It shows even if you increase much sequence depth, but it won't help you too much. And also the saturation point is quite important, but it will be very highly variable. It totally depends on the transcription factor, histone modification, or even the genome of your study. So it's totally case by case. Another thing we have to think about the ChIP-seq experiment is a chromatin fragmentation. As we showed before, it's in the first step, how we do fragmentation. So we have two ways to there. I should put at a slide to remind you. So two ways, one is a sonication, the other one is MNS digestion without cross-link. What's the difference of this one? And MNS can digest the link DNA between nucleosomes, right? Yeah. And somatic creation, they just fragment all the sequences around here. But still, case by case, depending on what you want to study. And then you may pick up which fragment you used. For example, if you want to study histone modification, definitely we don't want those link DNA region. So we can use MNS to remove those one first. Then we can do other purification or amplification, then sequence there. But of course, there's still disadvantage of this one. You have to be careful to use them when you use them. It's one thing is nucleosome, they are not always stable. Not all nucleosomes, they are stable. It's a dynamic process. Some nucleosomes, if they are not stable, they may be removed by this too. So you miss the signal here. But potentially, and this technology have some bias here. But the good thing is they can remove those ones. They may improve the sequence, like efficiency, focus on this part, remove those parts. However, when you study transcription factor, and what do you need? If you have a hypothesis or if you have knowledge about those transcription factor, you know those transcription factor tend to bind the linker DNA. Definitely, you cannot use this fragment technology. You have to use a sonication. Here is one example. They show two conditions, and this arginine receptor, they are binding site. And what they show is a binding peak. And you can see the first condition is fine, the most peaks here. But second one, why they see some distance here? Because they located on link region. Those are nucleosome regions. They located here. If you use MNS, then those true binding site will be removed. Then probably you will lose most of true binding site. That's not good for you. But when I talked about the true peak, so what do we mean peak? And I have to repeat that again. The peak means the sequence we captured and which are of what we're interested in. either transcription factor binding or ion binding or whatever open promoting. So those are what we catch. When we catch that, when you align the sequence on the whole genome, you may pair up those sequences in some area to see such enrichment. But can you call those peak as peak? Can you? Yes or no? Okay. Sorry. Yeah, go ahead. Yeah, looks good, right? Because when you call them peak, you compare with a neighbor or local region. They might be. But however, just to remind you again, for example, ChIP-seq, we use the antibody. We use a specific antibody to put that one. But some sequence, they don't have antibody. If we don't use antibody, we may get a pretty similar pattern in some area because those sequences, they tend to be caught up. So for example, like this one looks like without a comparison, and this one is stronger than that one, you say, okay, I like this one. This is a true binding site. But when you use a control, you use a naked DNA without antibody binding, and you do ChIP-seq, and you find there is a peak here, then you say, okay, those are false positive. And when you compare this one, you may make a conclusion those are true peaks. So let's bring us another consideration. That is, do we need a control? And we definitely, I prefer we need a control, but again, depend on what you are going to study. And there are three types of controls we can use. One is an input DNA, no IP. And another one is a mock-IP DNA, so IP without antibodies, or maybe non-specific IP. So IP performed using an antibody against a protein of your study. That is not well known or to be involved in DNA binding or chromatin modification, such as IgG or something. But again, do we really need a control? The answer is I prefer have control. Unless you can guarantee you have enough amount of sequence to avoid the sample in bias, but it's very difficult. Or maybe we can avoid control if we compare two conditions. For example, here, this is a control, but if it's not control, it's like if we call this is Y type and this is treatment, then after both Y type and the treatment, they will show this figure. Then they will diminish. Yeah, then we can say, okay, we get a difference. We only care about difference. Then maybe we don't need a control because both of them will need a substrate the control. So in that case, probably if you want to save money, you want to say something, it's fine. You don't need a control. So here is a two control comparison. It's an input or IgG. So it's basically they will process in the same way, but one is without the IP, and the one is using non-specific antibody as I showed previously. However, the input one without IP, they do not demonstrate flat or random distribution, especially some area like open chromatin region or promoter region or repetitive region, they tend to be fragmented and can be easily during the sharing and can be amplified and then mapping to the whole genome. That will result in the false positive. Then IgG is good. IgG is better. Hard to say because IgG may be less desirable in some circumstances. It's also case by case. Most IgG antibodies, they are not obtained from true primer serum from the same animals and in which the specific antibody was raised. And also IgG antibodies, usually the IP much less DNA than specific antibody do. And it's limited genome region from the control may be over amplified during the library construction step. So I don't ask you to know exactly what are those means in biological. Even myself, I don't know exactly what that means, but you just keep in mind when people are talking about the input control or IgG control, you have to know what's the advantage of those control, what's the disadvantage of those control, and what we are going to study. That's the most important thing. You have to understand what you need, what you want to do first. Then you can know what you need. Yeah. You want to take a 5 minutes break? Okay. Yeah.